The overall goal of this behavioral bioassay is to investigate odor-guided behavior in vinegar flies. Flywalk is a high-throughput system that tracks and analyzes the behavioral responses of multiple individual flies to olfactory stimuli automatically.
This new technique can help answer key questions in the area of insect olfaction, like what is the hedonic valence of an odor, or what does the addition of additional odorants mean to this valence?
The main advantage of this experiment is that we can examine odor-guided behavior in high temporal resolution, and we use odor stimuli that are similar to those that insects experience in nature, or to those that we also use in electrophysiological recordings. Generally, individuals who try to use this method might struggle because it needs an airtight system. So you need to know where the air is going through, and you need to be sure that there are no leaks at all where the air can leave the system.
For these experiments, it is important to rear the flies under a 12-to-12 light-dark cycle in low to mid-density cultures containing a normal food medium. Keep the room temperature between 23 and 25 degrees Celsius, and the relative humidity should be around 70%. In preparation, using large culture vials, mate 20 to 30 adults for a week, and collect groups of 30 to 40 offspring of mixed sex to make an experimental cohort. When a cohort is 2 to 4